ये ऊपर है
ये सामने रहेगा ना, ये यहाँ रख, ऐसा रख, ऐसा रख।
नहीं नहीं बैक पे आ रही है,
फ्रंट कैमरा यूज करना,
फ्रंट कैमरा यूज कर ऐसा रहेगा।
हाँ ऐसा रहेगा कोई। सर जी उनको बोलो क्या,
अब लगा के देखो सर वो हाई
कब से अब वो ऑन ही नहीं कर रहे।
Everyone. Good morning.
ऑडियो
ऐसा होता क्या?
गुड
Good
मॉर्निंग
morning. ये सबके माइक ऑफ कर.
गुड मॉर्निंग गुड मॉर्निंग गुड मॉर्निंग
Good morning.
This is echoing.
Good morning. Morning. आवाज येतो बघा.
रजिस्टर आनंद डायरेक्ट
दो आपका सबका माइक ऑफ करो और ध्यान से. सबके माइक ऑफ करो. हाँ. चलो बंदा. Good morning. Good morning, everyone.
मी पण येतो. गुड मॉर्निंग. नहीं अभी किया है.
Hey,
हेलो 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 हेलो
हेलो. हाँ हेलो.
ठीक है ना इसमें बैकग्राउंड ठीक है क्या ये दिख रहा है
जादू सर सर्वांना म्यूट करा.
Hello? Am I audible? Bismillahirrahmanirrahim. The chief guest of this inaugural session, Dr. Shamama Parveen, President, Raibar Educational, Cultural and Welfare Society, Professor Surendra Singh, the keynote speaker, the galaxy of intellectuals, professors, researchers, and my dear students. A very pleasant good morning to all of you present in this inaugural function of this one-day national seminar on recent trends in plant sciences, organized by the Department of Botany. Let's begin the program with the recitation of Quranic verses. I invite Dr. Muhammad Mustafa Khan, the head Department of Arabic, to perform Qiraat. Dr. Muhammad Mustafa Khan.
أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. فلينظر الإنسان إلى طعامه أنا صببنا الماء صبا ثم شققنا الأرض شقا فأنبتنا فيها حبا وعنبا وقضبا وزيتونا ونخلا وحدائق غلبا وفاكهة وأبا متاعا لكم ولأنعامكم فإذا جاءت الصاخة يوم يفر المرء من أخيه وأمه وأبيه وصاحبته وبنيه لكل امرئ منهم يومئذ شأن يغنيه وجوه يومئذ مسفرة ضاحكة مستبشرة ووجوه يومئذ عليها غبرة ترهقها قترة أولئك هم الكفرة الفجرة صدق الله العظيم. سامعین اکرام، قرآن کریم کی چند آیات آپ کے سامنے پیش کی گئی ہیں. ان آیات میں نے جہاں سے انتخاب کیا ان آیات میں اللہ رب العالمین نے نباتات اور انسانی ضرورت کے لیے جن غذاؤں کا ذکر کیا ہے خاص طور پر اس کو بیان فرمایا ہے. اس سے پہلے تخلیق انسان کو پیش کیا کہ انسان کی تخلیق ایک خاص مقصد کے تحت عمل میں آئی، اور پھر اس کے بعد یہ اللہ رب العالمین نے فرمایا کہ انسان کو چاہیے کہ وہ غور کریں کہ جو ہم نے اس کے لیے کھانا دیا ہے، وہ جو کھانا کھاتا ہے، اگر اسے کسی اور چیز پر غور و فکر کی فرصت نہیں ہے تو کم سے کم وہ چیز جو وہ استعمال کرتا ہے اس پر غور کریں کہ کیسا ایک سسٹم اللہ رب العالمین نے اس کی غذا کا پیدا فرمایا ہے. اللہ فرماتے ہیں انا صببنا الماء صبا، ہم نے بارش کے برسنے کا ایک پورا انتظام بنایا. یہ انسانی طاقت نہیں ہے بلکہ یہ اللہ ہی کی قدرت ہے کہ سمندر سے بھاپ کا بننا، فضاؤں میں اڑنا، پھر اس کا بادل میں ڈھلنا اور پھر اس کا برسنا، زمین میں اس کا جمع ہونا، جمع ہونے کے بعد میں پھر پھول پودوں اور درختوں کو اگانا. یہ کسی انسان کا کام نہیں ہے، یہ ایک زبردست طاقت کا کام. ہم نے پانی برسایا. ثم شققنا الارض شقا, پھر زمین کو ہم نے پھاڑا, اس میں سے بیج کو اوپر نکالا, اس میں سے پودوں کو اوپر نکالا, ہم نے اس کو چیرا پھاڑا. فانبتنا فیہا حبا, اور ہم نے اس میں سے اناج کو اگایا, ہم نے دانوں کو زندگی عطا کی اور اس میں سے پودوں کو باہر نکالا. یہ ایسی طاقت اور ایسی صفت صرف اس ایک ذات میں ہے جو سارے انسانوں کا پیدا کرنے والا ہے. اللہ نے ایک جگہ فرمایا کہ وہ فالق الحب والنوا ہے. دنیا کی پوری طاقت مل کر ایک بیج کو پھوڑ کر اس میں سے پودے کو باہر نہیں نکال سکتی, یہ صرف اسی کی طاقت ہے. اللہ فرماتے فانبتنا فیہا حبا, ہم نے اس سے اناج اگایا, وعنبا انگور, وقضبا اور ترکاریاں ہم نے اس سے نکالی, وزیتونا زیتون کا تیل اور اس کا جو درخت ہے, اس کا جو پھل ہے, اس کو بھی جو ہے تو ہم نے دیا ہے. ونخلا, کھجور کے درخت ہم نے پیدا کیے. اب اگر انسان یہ سوچیں کہ یہ تو بہت معمولی چیزیں ہیں کہ جس کا ذکر پیدائش میں ہو رہا ہے, تو اللہ فرماتے نہیں, وہ حدائق, جتنے باغات ہیں اس روئے زمین پر اور باغوں میں جتنے درخت اگنے والے ہیں وہ بھی ہم نے ہی پیدا کیے. غلبا, گھنے باغات, ایسے نہیں کہ باغات میں چند درخت ہیں بلکہ گھنے باغات ہم نے اگائے ہیں. وہ فاکہہ, جتنے پھل اس روئے زمین پر پائے جاتے ہیں وہ ہم نے اگائے ہیں. وہ ابا, گھاس ہم نے پیدا کیا ہے جو جانوروں کے لیے کھانے کا ساز و سامان ہے. اس کے بعد اللہ فرماتے ہیں متاعا لکم, یہ تمہارا سامان ہے, تمہارے لیے یہ دیے ہیں. ولانعامکم, اور تم جن جانوروں کو استعمال میں لاتے ہو اس کی غذا بھی ہم ہی نے اگائی ہے. تو یہ آیات جس میں اللہ تعالیٰ نے اس بات کا ذکر فرمایا کہ اس کائنات میں ایک چھوٹے سے چھوٹا پودا اور بڑے سے بڑے درخت تک کو اگانے کی ہم نے جو نظام بنایا ہے, جو سسٹم بنایا ہے, یہ انسانوں کی اپنے استعمال کے لیے ہیں, ان کی بھلائی کے لیے ہیں. اس کے علاوہ قرآن کریم میں جن پودوں کا ذکر ہوا ہے اس میں العنب, انگور کا ذکر ہوا, عنب و قصبہ, ترکاریوں کا ذکر, الرمان, انار کا ذکر اللہ نے سورہ رحمان میں اور اس کے علاوہ اور کئی جگہ فرمایا ہے. فیہا فاکہۃ ونخل ورمان, زیتون کے درخت کا ذکر ہے, کھجور کے باغات کا ذکر ہے, کھیتی میں اگنے والے تمام چیزوں کا ذکر ہے. وزروع ونخل طلعها عظیم کہہ کر اللہ نے اس طرف اشارہ کیا. رطب, کھجور کی کئی ساری اقسام ہیں. ایک کھجور جو جسے ہم اردو میں ادری گدری کھجور کہتے ہیں جو بہت مفید بھی ہوتی ہے, اس کا بھی ذکر اللہ نے رطبا جنیہ کہہ کر قرآن میں بیان فرمایا. فاکہہ کہہ کر پورے پھل جتنے انسانوں کے صحت اور استعمال کے لیے وہ کھاتا ہے سب کا ذکر ہے. زیتون میں زیتون اور اس کا پھل ہے, اس کا درخت ہے, اس میں انسان کے لیے فوائد. ایک لفظ استعمال ہوا ہے ثمر, جو عام پھلوں کے لیے استعمال ہوتا ہے, وہ بھی اس کے بھی درخت کا ذکر قرآن میں ارشاد ہے. یقطین, دودھیا جسے اردو میں کہتے ہیں, اس کا ذکر قرآن میں ہے کہ اللہ نے اس کا بھی درخت پیدا فرمایا. السدر, بیری کا ذکر ہے. ککڑی, دالیں جتنی ہیں سب کا ذکر ہے. لہسن ہے, پیاز ہے, اور بالیاں ہیں, گیہوں کی بالی کا ذکر قرآن میں استعمال ہوا. پھول, خوشبو والے جتنے پودے ہیں ان کا ذکر ہے, اور ریحان کہہ کر. زنجبیل, سونٹ, پھر درخت شجر کہہ کر جو ہے پورے درخت کو شمار کیا گیا. اب نباتات میں جتنی چیزیں زمین سے اگتی ہے, اس طرح سے قرآن کریم میں اللہ رب العالمین نے ہم انسانوں کو غور و فکر کی دعوت دی ہے کہ جو رب تمہارے لیے تمہاری زندگی گزارنے کے لیے اتنی چھوٹی چھوٹی چیزوں کا خیال کر سکتا ہے, اس کا کارخانہ کتنا عظیم الشان ہے اور یہ کائنات کتنی بڑی ہے. اللہ تعالیٰ سے دعا ہے کہ اللہ تعالیٰ ہم تمام کو قرآن کریم میں جن تعلیمات اور جن سائنس سے متعلق جن چیزوں کا ذکر ہے اس کو جو ہے تحقیق کرنے اور اس سے ہمارے لیے اس کو مفید بنانے کی توفیق عطا فرمائے. اللہ نے سچ فرمایا, جو کچھ بھی فرمایا.
تھینک یو ویری مچ سر. فرصت کہاں تھی اتنی کہ ہم کھولتے کتاب, فرصت کہاں تھی اتنی کہ ہم کھولتے کتاب, ورنہ تمام علم تو قرآن ہی میں تھا. We are happy to announce that the participants from various states got their names registered in this national seminar. On behalf of Sir Syed College of Arts, Commerce and Science, Aurangabad, Maharashtra, India, I, Dr. Sheikh Muhammad Azhar, extend a cordial welcome to you all. The eminent scholar, the educationist, the loving teacher, and the true leader, late Professor Muhammad Tilawat Ali is the founder of Sir Syed College. Undoubtedly, Sir Syed College is the outcome of untiring, devoted, and the selfless efforts of Professor Muhammad Tilawat Ali. Sir always believed that education is meant for all-round development. Let me give the brief introduction of Sir Syed College. Sir Syed College of Arts, Commerce and Science is the minority institution in Aurangabad city, established in 1990. It is one of the leading institutions engaged in imparting quality education in the stream of arts, commerce and science for the last 3 decades. The devoted and committed leader known all over Maharashtra for his selfless services, late Professor Muhammad Tilawat Ali, established Sir Syed College that could meet the demands of changing times. Professor Muhammad Tilawat Ali named this college after the legendary Sir Syed Ahmad Khan, an optimistic social reformer and visionary. It is in fact the only co-education college in the town that has largest number of girl students. The ongoing century, being the century of youth, the college is marching ahead under the able guidance of the President, Rehbar Educational, Cultural and Welfare Society, Dr. Shamama Parveen. And we are quite confident that the new dynamic and positive thinking of Dr. Shamama Parveen will lead the institution from the front and take the institution to the appreciable heights. The college offers arts, commerce and science at junior and undergraduate level, postgraduate courses in Arabic, Urdu, English, commerce and organic chemistry are also taught here. To meet the changing economic challenges, the college also offers 3 years professional degree courses such as BBA and BCS. The institution always takes the initiative to prove its mission, humane citizen through education. The college has a team of 34 experienced, well-qualified and dedicated faculty with 13 research guides in Arabic, English, commerce, history, chemistry, botany and zoology. 29 teachers are doctorates and 11 teachers have qualified next exam. Under the NSS, the college is involved under various social activities. Sir Syed College is the only college in the region winning intercollegiate football tournament consecutively for 7 years. The faculty is also involved with the research work through minor and major research projects. The dedication and teamwork of teaching and non-teaching staff has won several laurels to the institutions. 34 students so far have stood in the merit list of Dr. Baba Saheb Ambedkar Marathwada University, Aurangabad. 3 students received gold medals in arts and science streams. Today, approximately 2,000 students are on the campus of Sir Syed College. 2 commerce students of our college have qualified CA exams. Hundreds of students are regularly placed in companies and different offices. 4 students of our junior college got admission for MBBS and BDS courses. In the recently declared results, Sheikh Nausheen appeared 9th in the Dr. Baba Saheb Ambedkar Marathwada University merit list, and Khan Asma scored the highest marks in zoology and became eligible to receive Miss Anita Awad Award. We are all bound to dedication, quality, and excellence. Dear friends, we are quite fortunate to have Professor Surendra Singh, Professor, Department of Biological Sciences, Rani Durgavati University, Jabalpur, Madhya Pradesh. He is available as a keynote speaker. So I request Professor Surendra Singh to declare the formal inauguration of this one-day national seminar. Professor Surendra Singh.
Thank you. Thank you so much. First of all, the respected chief guest, Dr. Shama Parveen ji, and the president and the principal of the college, Dr. Sheikh Kabir Ahmed ji, the convener of this one-day national seminar, Dr. Milind Jadhav ji, and the co-convener, Dr. Sangeeta Ahuja ji, and coordinator, Dr. Sangeeta Sutar ji, and all other the member of the organizing committee. I congratulate the college for organizing such a vital one-day national seminar on recent trends in plant sciences. Because the plant is playing a very important role, as just have been said in the Quran itself, the significance of the plants in the human life, which has been created by the God and the Allah on this earth. So organizing such a vital national seminar by the college, I really congratulate all the members of the faculties of and the organizing committee for having this seminar and inviting me today to share some of our few words with the participants. Without taking much time, I declare this national seminar inaugurated and open. Thank you so much.
Thank you very much, sir. We would definitely love to listen a lot from Professor Surendra Singh in his keynote address after a short while. Respected participants, I would now like to invite Dr. Milind Jadhav, the convener, head and IQAC coordinator, to tell the aims and objectives of this national seminar. Dr. Milind Jadhav.
Good morning, distinguished academicians. Good morning, distinguished academicians, researchers, students. Today, I am here to welcome all the researchers, dignitaries, and students to this conference, to this seminar. Respected participants, on this occasion, we would like to take this opportunity to pay a tribute to founder of our Sir Syed College, Professor Muhammad Tilawat Ali sir. He was a constant source of inspiration and encouragement. He used to get actively involved in all academic activities. We are really missing him here. May his soul rest in peace. I would like to express my heartfelt thanks to the chairperson of our college, Dr. Shamama Parveen ma'am, and our principal, Dr. Sheikh Kabir Ahmed, for their support and guidance to organize this seminar. I am also thankful to Professor Surendra Singh, Department of Biological Science, Rani Durgavati Vishwavidyalaya, Jabalpur, and Professor Shailendra Jadhav from Pandit Ravi Shankar Shukla University for as a chief guest. Seminar is the academic gathering of stalwarts, scientists, academicians, teachers, researchers, and students. It is the place of sharing and exchanging thoughts and ideas. Plants are important to all life of Earth. Research in the field of plant sciences contribute directly to the economy and development of nation. The result of research are beneficial to human being. The main objective of e-seminar is to know and understand the recent researches in the field of sciences. Today, in whole day, we will have a very good discussion and sharing of knowledge regarding recent trends or recent researches in the field of plant science, particularly applied aspects of the research in plant sciences. With these few words, I would like to call Professor Muhammad Azhar sir.
Thank you very much, sir. I pay my sincere thanks to Dr. Shamama Parveen to praise the occasion. We are thankful to Professor Surendra Singh for making the formal inauguration. We are thankful to you for your valuable presence, sir. We are grateful to Dr. Sheikh Kabir Ahmed, the principal for his guidance. This inaugural session is immediately followed by the keynote address by the legendary Professor Surendra Singh from Jabalpur. Now I request Dr. Milind Jadhav to introduce Professor Surendra Singh and proceed. Thank you.
Professor Surendra Singh sir, Professor Shailesh Jadhav sir, and all distinguished researchers, academicians, teachers, and students. Before introduce Professor Surendra Singh, I would like to call Professor Shailesh Jadhav sir as a chairperson or chairman for this keynote session. I would like to introduce Professor Shailesh Jadhav sir. Shailesh Jadhav sir is working as a professor in School of Studies in Biotechnology, Pandit Ravi Shankar Shukla University, Raipur, Chhattisgarh. Professor Shailesh Jadhav also worked as a former controller of examination, Pandit Ravi Shankar Shukla University, Raipur. His area of research is mycology, aerobiology, and bioenergy. 52 MPhil students and 25 PhD students awarded and completed MPhil and PhD under his able guidance. Sir has published 152 research papers in national and international journals. 6 research projects from different funding agencies have completed by Professor Shailesh Jadhav sir. Jadhav sir has organized 10 seminar, conference, and workshop. Professor Jadhav sir has written 9 books on different aspects of biological science. Now I would like to introduce today's keynote speaker, Professor Surendra Singh sir. Sir is holding post in Rani Durgavati University, Jabalpur. He is the director of University Institute of Vocational Studies, Director UGC Community College, Coordinator Distance Education System Guidance and Counseling Center. Sir is also having a charge of Nodal Officer, Skill. Sir is also a chairman of Central Board of Studies in Botany, Government of MP. Sir is Commonwealth Postdoctoral Fellow of University of Duneep, Scotland. He is a visiting scientist, Jacob Goldstein Institute for Desert, Israel. Sir is also visiting scientist to the Institute of Hydrobiology, his Academy of Sciences, Wuhan. Professor Singh sir have a PG teaching experience of 31 years, research experience 38 years, supervised 8 research projects. Under his able guidance, 12 students completed MP and 16 students completed PhD. Sir has published 60 research papers in national and international journals. His research area is hydrobiology and biotechnology. Sir is also mainly associated with different pharmaceuticals and biotechnological company as an advisor. Sir has established, recently sir has established a biotechnology lab, Trikal Biotechnology Lab, and through this Trikal Biotechnology Lab, that biotechnology lab is organizing a number of many programs, awareness programs, and regarding especially organic farming. With this brief introduction, I like to call Professor Surendra Singh to deliver a keynote address.
Thank you, Dr. Milind Jadhav ji. Thank you so much. First of all, I want to thank all the member of the organizing committee and college management, especially you, sir, for having invited to this a very significant national seminar on recent trends in plant sciences. So today I'm going to speak on the algae and human welfare. So let me share my screen first. Share screen. Then. सर ये दिख रहा है आपको स्क्रीन? हेलो?
जी सर.
Yes, sir. Yes.
Yes, sir.
Okay. So this is my area of specialization from Masters from Banaras in the University on algal biotechnology. And so I started with the Masters in a specialization in algal biotechnology. And since then, I have been working on the various aspects of microalgae for human welfare. As you know that most of the people, means those who are associated with the botany, they know the importance of lower plants, because now during this pandemic time, everybody is talking about the role of medicinal and aromatic plants in combating this pandemic of COVID-19. And a lot of discussion is going on that our Ayurveda and other, that how these plants, particularly those medicinal plants, are playing an important role in boosting the immunity of our body. So I would like to tell you that it is not only the higher plants which can play a very important role in boosting the immunity of our system, but there are lower plants also, particularly the under the thallophytes, the algae, the shaival, the kai, which plays a very also important role in the improving the immunity of our system. So most of us, we don't know actually how the algae can play an important role in human life, because we think that it is a waste things, and polluting the water here and there. And if you see that there are more than 25,000 of species of algae is there. Most of the algae is growing in the sea because they are the marine algae, and also in freshwater systems, so they are freshwater algae. So we can classify the marine algae and the freshwater algae. And if you look, because they contains variety of pigments and other secondary and primary metabolites, let me tell you the most important contribution of algae in human life is the production of oxygen, because they are only involved along with the higher plants oxygenic photosynthesis, because they evolve oxygen during the photosynthesis. So 70% of this oxygen, prana vayu, comes from algae. Only 30% or 30 to 35% comes from the higher plant, because only this much of plants and higher plants and the forest are left on this earth. So 70% the prana vayu, on which we survive, the whole humanity survives every day. We inhale, oxygen comes from the algae. So that is the most important, and they are the responsible, particularly the blue-green algae, the cyanobacteria, for making this earth oxygenic, because earlier environment of the earth was anoxygenic. There was no oxygen. So they are the first primary introducer of oxygen on this earth is the algae, and that is the blue-green algae. So in a simple sense that we should understand this prana vayu, the oxygen, जो इस धरती के ऊपर है, जिसके ऊपर हम जीवित हैं, इसका 70% भाग एल्गी से आता है, and 30% from the higher plants. So that is the most important role of algae in our life. So now there are the various groups of the algae. Those in the plant sciences, they know the cyanobacteria, the chlorophyta, the rhodophyta, brown algae, and so many groups of the algae are there. If you look the types of algae, mostly this red algae, this macroalgae, they grow in the seawater. So most of the algae which grows in the seawater, in the marine water, they are playing a very important role in human life as a food, as a feed, as a source of cosmetics, as a source of fertilizer, and as a source of secondary and primary metabolites, even those agar-agar which we are using in a laboratory, that mostly comes from the red algae. So, and particularly the countries, Far East countries, they are using this seafood, algae as a seafood, day-to-day life. We are not fond of that in India because it has not been introduced properly in our food system. But if you look here, the China, the Singapore, the Malaysia, the Japan, and the Russia, and other countries, they are mostly dependent upon the algae as a food and feed and so many things. Though in our country, there are institutions, the government institutions, they are working on that and they are producing a lot of things out of that. But there is an urgent need also to emphasize the role of this marine algae in human life as a food, as a feed, as a cosmetics, as a fertilizer, and a source of various metabolites. That is important. So then these are the various photographs of the microalgae. Even 70 meters length of this seaval is grown into the seawater. About this microalgae, the small one, the smaller in size, which generally we grow here because we are far away from the sea. So mostly we are working on the microalgae, the cyanobacteria, the green algae, which are smaller in size, but they also play a very important role in human life and the human welfare. And they are the source of the products like, carotenoids, antioxidants, fatty acids, enzymes, polymers, peptide, toxin, esterol, whatnot. They are playing very, very important role. So now I'm just showing the some of the commercially well-known members of the microalgae, which has been commercially been exploited. Let me tell you, because being a student of Banaras Hindu University, where the School of Algology was established, and that is the Center for Advanced Center for Algae and Ecology, and now the Institute of the Science. Last year, the centenary celebration was there, 100 years of the Banaras Hindu University and the department. So I asked even that though there are great scientists and the great teachers with all due respects, but whatever the researches has been carried out, that has not been commercially exploited. If you look at the role of Banaras University, they are working on physiology, biochemistry, the genetics and the molecular, but none of the technique has been developed and commercialized. The mass cultivation of economically important microalgae is important. Because we grow in the laboratory in very small scales in 5 ml and 10 ml, 20 ml and 25 ml is not going to solve the industrial solution. So there are the commercially known members like spirulina. Everybody knows that spirulina is the blue-green algae and the richest source of protein on earth. Then we have a Dunaliella, that is the richest source of beta-carotene. We have green algae Hematococcus, which is a source of astaxanthin. We have a Buttricococcus at the same time, is the richest source of oil and fatty acid. And then Nostoc, which is responsible for nitrogen fixation. So what I'm trying to say, you can see the nature has provided a lot of things to us. We have to identify, use, optimize, and utilize. You can see that spirulina is a blue-green algae that contains 71% of protein, whereas this Buttricococcus has got 70% of the oil at the two ends for the meeting the problem of bioenergy. So the microalgae all over the world, that is being commercially exploited at very large scale. $1 trillion market is there. But in India, we are unable to exploit the potential of microalgae. And I think this is the time when the Atmanirbhar Bharat Abhiyan is going on. We must concentrate and focus on these commercially important microalgae for exploiting the potential, vast potential, so that we can add in the economy of our country. Now, if you look here, the algal biotechnology, what algae can do in our life, algae as a food, as a feed, as a cosmetics, as a pigments under markers as a biofertilizer in agriculture, as a bioactive compounds, the pharmaceuticals, the biodiesel, bioethanol, biobutanol, everything is there. If you name, so algal biotechnology aims at the exploitation of various genera of microalgae for commercial utilization. Now, I will just show you one by one. Look here, the algae as a food. Mostly the chlorella, which has been green alga, has been exploited as for the tablets and the food, and for also the antibiotics. Chlorelline has been extracted from the green alga chlorella. And then most important is the spirulina, the richest source of protein on earth. You can see the lot of food products have been developed from these spirulina, the tablets and the chickpeas and the juices and the sauce and whatnot. Whatever you want to prepare, it is there. So as you can use the algae as a food, the Central Food and Technological Research Institute, the CFTRI, they have developed the technology of spirulina cultivation, production, and all those things. Now, algae as a food products, if you look here, you can prepare this ice creams, the chocolates, the drinks, the papad, and the biscuits, and whatever you want, you can develop. And this technology has been developed in our country also, not exploited. That is the problem. What I'm trying to emphasize, there is a need for exploitation of these resources for economic development. Now, if you look here, the comparative data for protein content in the different algal species, you look here at the Spirulina platensis, the Spirulina maxima has got 71% of the protein. So this is the richest source of protein on earth. 71% out of 100 grams, you will get 71 grams protein, and that is vegetarian protein. Anybody can use it. There is no discrimination that it is animal protein. Somebody thinks that animal protein they will not consume, but this is a plant protein. So and the richest source of protein. I would like to tell you that whatever the sources are present on this earth of the protein, if you look at the I will show you some slide where you can see that if they are the other sources like milk has got 2.5 to 3% or 3.5% of milk protein out of 100 grams. So we are emphasizing every day to drink milk. What for? Only 2% of the protein. That is, if it is pure, it's not added with the water. So only on 2 to 5% and 3%. Then egg contains around 11% of the protein. The pulses, they have got 20% to 25 to 30% protein. Soybean has got 35% of the protein. The mushroom has got 45% of the protein, but the spirulina has got 71% of the protein. So we have to think whether we should go for milk or we should go for spirulina, which has got 71% protein, but the milk has got only 2.5% of protein. So what changes in our food habit has to be made, that has to be emphasized. That's what I said, that it has not been actually popularized among our masses from the beginning, that these are also the sources of various proteins and other important metabolites for our life. Now, if you look here, the carbohydrate content, if you look at the carbohydrate content, the spirulina, other things, they have got only 14%. So those who are diabetic in nature, they can take spirulina safely because the carbohydrate content is very low in that. Look here, the lipid content. Again, the spirulina content has got here, you can say the 3% as compared to other algae. So it is safe, but it's not going to increase the cholesterol level in our body. So therefore, the spirulina can easily be taken by these people who are either suffering from diabetes or obesity. For them, the spirulina is the safe thing. Now here I am giving this data, that comparative data for the protein content of the different protein sources. You see out of 100 grams, here you can see the milk has got only 3.5%. The cheese has got 27%. egg has got 12%, the apple has got 0.2%, 0.2%. Save apple, every time that eat a apple keeps the doctor away. So only banana 1.7%, the potato 2%, the bread white 4.8%, fish 20%, oyster 13%, beef, potato, pot rest 22%, liver 23%, and whatever, the pork, the ham, the peanuts butter, the pecan, all those things. You will look carrot, only 1%. Look here, the algae has got the 71% of the protein content. So highest content of protein is present, and that is digestible protein. It can easily be digested in our stomach system, is the algal protein. Now, if you look here the comparative data for vitamin A, you can see here the again this is the algae, the red one. You can see that 1,80,000 mcg as compared to other sources like ghee has got 700 and something like that. If you'll see the chili powder, these mangoes and all kind of spinach, they have got very less amount of vitamin A, where this algae has got the highest source of vitamin A, which is responsible for our retina, for our brain development, for whatnot. Every time we say that we should take carrot for vitamin A, you can look here the carrot. I think this year only had got 8,000 mcg out of 100 grams as compared to 1,80,000 in algae. So the content I'm trying to emphasize that if you look here the different kind of vitamins, and this is again the vitamin B12, because this is responsible for our maintenance of our heart. Is the rhythm. Whenever there is a heart attack, vitamin B12 injections are given to restore the activity of the heart muscles. And vitamin 12 is playing a very important role. So you can see the milk has got only 0.4 mcg of vitamin 12 when we are emphasizing more on the milk as compared to 100 here in the algae. So there are the egg only 5. So there are n number of food stuffs which has got very less amount of vitamin B12, where the algae has got the vitamin 1. At the same thing, if you look here, the different sources, the potato, the kidney beans, the pork, the wheat, the everything, the algae has got the highest source of vitamin A. So this is again the comparative data for protein content, which I have already discussed. Leucine, important, the amino acid. The algae again has got the highest amount of the leucine here. The other protein, look here, lysine also. Algae has got highest amount of amino acids. This isoleucine, again, if you look here, the milk has got only 2 mg as compared to algae. This is again the algal protein amino acid profile. You will find that algae is dominating in all the spheres. This is the spirulina which we are talking about. Why it is called the spirulina? Because it's a spiral in shape. There, just like a spring, therefore it is called spirulina, and it was originated 0.5 billion years ago on this earth. And the best thing is that spirulina grows at above pH 9. So the other microorganisms cannot compete because it's highly alkaline in nature, alkalophilic. So others, that's why this spirulina can be grown outdoors in a large quantity, and where it can compete and survive well in outdoor open system because of the high alkalophilic nature of the spirulina, because it grows beyond 9 pH. Now, if you look here, the historical background of the spirulina, which has been utilized by the tribals and particularly the South American countries, and in Lake Chad, in the country Chad in Africa, they have been utilizing spirulina without knowing that this is the richest source of protein on earth. This is the nutritional composition. If you look here, again, the protein, the carbohydrate, the moisture, the minerals, and the fats, spirulina is the best. This is the pigment composition. Why it is called blue-green algae? Because the phycocyanin pigment dominates here as compared to the phycoerythrin, because they are the ancillary, the accessory pigments present in the blue-green algae. That is why it's called Neel Harit Saiwal. The green color is imparted by the chlorophyll, where the blue color is by this accessory pigment that is called the phycocyanin. Because phycocyanin dominates in blue-green algae, therefore they are called blue-green algae, whereas in the red algae, phycoerythrin dominates there, and therefore they are red in nature. But both the pigments are economically and medically very, very important. You look here, again the spirulina, the different important role of the spirulina, because they're rich in iron, the magnesium, the trace minerals, healthy for nerves and the tissues. 60 to 70% of protein, high source of vitamin B12. It is also important role plays in eyes and vision, build the muscles, increase the stamina, healthy digestion, natural cleanser, and they also removes the toxins, the free radicals, rich in gamma linolenic acid, which is very important. GLA plays a very important role in anti-aging, and then also it prevents the cancer, which is very important because the presence of the pigment phycocyanin. The phycocyanin, the blue pigment present in the spirulina and the blue-green algae as such, is anti-cancer in nature. The anti-cancer property present in only in this pigment of blue-green algae as spirulina, that is in phycocyanin. Now, if you look here, these tribes in the Lake Chad, one of the African country, where the spirulina grows naturally in the lake. And that is the belief of the tribals that men will not go into the lake, otherwise the goddess of the lake will be angry. So I don't think why they are showing, but they are sending their ladies only to collect the harvest the spirulina. They don't know what they are eating, but they have been eating since long, since the civilization. So first this leader lady goes into the lake and collects the spirulina culture and then brings out here, you can see, and but because they don't have any filter, because the high temperature there, sand is there, so they are pouring on the sand so that the water goes down, and when it get dried up in the sun heat, you can see here, they are collecting it and selling in the market and utilizing as a food. So that is their routine. They have been using and utilizing unknowingly, because that's why the scientists, when they have seen that what they have been eating out of this lake, and they are hale and hearty and they are very good in health, then only they have been the spirulina has been identified. Oh, this is a richest source of protein on earth. So these tribals, they have been using this. Now, this is the photograph when I was heading the company Hash Biotech Labs Private Limited in Chandigarh. As a chief technology officer, the technology was developed and given by us, me only. So here you can see that the scaling of the spirulina, here the outdoor, the cultivation in Mohali, that spirulina was cultivated like this. And then this is the production center I have developed in district Hoshiarpur, the large scale cultivation of spirulina. And you can see here around this spirulina ponds were in around 8 to 10 acres of the land. So more than 150,000 liter culture was maintained by us. And then harvesting, and then it was converted into tablets and all other products. You can see here, this is again the raceway ponds for cultivation of spirulina in Mohali. And this spirulina was used as a pledge campaign, global spirulina campaign for malnutrition, hunger and poverty. Here I want to emphasize you that the United Nations in 1974 itself has declared that spirulina is a superfood for tomorrow. And there is a intergovernmental institution called IMSAM, IIMSAM, Intergovernmental Institution for Use of Microalgae Spirulina Against Malnutrition. So that is the mandate of the United Nations, for using spirulina as a key driver to eradicate malnutrition, because of the malnutrition, every 7 seconds one child is dying in the world. And India on the top of the malnourished country, and among the India, the Madhya Pradesh is on the top. So we have to fight this problem of the malnutrition by cultivating and distributing spirulina as a food supplement, as a key driver to control the malnutrition and then other problems. Now, this is again the effective eradication of malnutrition by using this spirulina. So because they are the source of the calcium, magnesium, phosphorus, vitamin, and therefore it is given to the this one. Then algae, spirulina as a algae to restore our planet. The benefits of the algal consumption, that the beta carotene helps fight some specific type of cancer and cardiovascular disease, because they are the source of beta carotene, the various microalgae, and they are antioxidants. Now everybody is talking antioxidants, that they are the natural antioxidant. There are a number of microalgae, not only the spirulina, but Dunaliella, the Haematococcus, and whatnot. The porphyridium, they have got the pigments and they have antioxidant property. So antioxidant means anti-cancer property, and they help in better digestion and for the growth of a nail, hair, skin, and then chronic diseases are controlled. Benefits of the algal diet is the gamma linolenic acid. I was talking about the GLA. The GLA is not synthesized and manufactured in our body. It has to come from the other sources. Therefore, in the newborn babies, the breastfeeding for 6 months from the mother is required, because the GLA comes from the mother milk to the newborn baby, and that GLA is converted into omega-3, omega-6 fatty acid, and which is the precursors of various hormones, and also they are the precursors of the brain, the nervous system, the heart, inner layering. So it plays a very, very important role for development of the cognitive development. And also it provides the immunity to the child. So the gamma linolenic acid is very important. Therefore, the fishes which are feeding on the algae, they are rich in gamma linolenic acid, and they play an important role in the development of the brain and eyes and all those things. So this is an immunity booster, and that is also a source of alkaline food. Now, algae can also be used as a feed, and here I will just want to show you. This is the photograph when I was working as a visiting scientist in Jacob Blaustein Institute for Desert Research, Ben Gurion University of the Negev in Israel, 1.5 years. Now, you can see this is Professor Amos Richmond, the founder director of this institute, and one of the leading scientists in the world on microalgal biotechnology. And he has taught me how to cultivate microalgae, particularly red microalgae porphyridium. Here you see, this is my photograph in 1998. I was standing with Professor Richmond, and I have grown this red microalgae using flat plate glass photobioreactor. This is also flat bioreactor. Here you can also see what we have been growing here. This is a flat bioreactor, 1,000 liter of the culture. Nanochloropsis was grown, and this Nanochloropsis or Isochrysis galbana is used for the aquacultures, the feed for aqua animals, and they are rich in EPA, eicosapentaenoic acid, and arachidonic acid. This eicosapentaenoic acid that prevents the heart diseases, asthma, cancer, even the menstrual disorders, hay fever, and also lot of liver dysfunctions. That is why the Professor Richmond was developing this technology, cultivating Isochrysis galbana, this Nanochloropsis, and other microalgae for these kinds of fatty acids and other metabolites. So they are actually exploiting. They have sold the technology to a Swedish pharmaceutical company in 1990. In $1 billion deal was there. So they have been, actually the Israelis, they know how to exploit any natural resource for the commercial purpose, and they are gaining. Because Amos Richmond, you can see, they are the Chinese scientists, they are working with me. This is a German technician was also with me. This is Professor Richmond. He was saying Dr. Singh is a solar biotechnology. We are working on dollar biotechnology. How freely available solar energy can be exploited into, can be converted into dollars. That is how we are working. We are working on solar dollar biotechnology. You can see the concept, because they are not having much, but they know how to exploit the resources. That is why their technology, because so small country, we should take examples and which from the Israel, because it's only 350 kilometers long and 75 kilometers wide country. Only 60 lakh people are there. 3 lakh people, less than the population of any other state, and is smaller, but they are the world leader in all the technological aspects. Their dry land irrigation system, their drip irrigation system, we are all following, and they are leaders in technology. Therefore, cutting edge research technology is required for any country to be developed and to go ahead. Therefore, it is very important for all of our those scientists and teachers and the researchers also, that we should not compromise with our country. Country is nation first. We must work on that, on researches. True researches, cutting edge technological researches are required at the moment to make this Bharat as Atmanirbhar. So this I have learned from Professor Richmond. Now you can see the algal feed is there, the algal products for various aquatic animals, and the algal diet for aquaculture, you can see for fishes and for and for other things. They are providing immunity to the animals. You can see that this is also a feed for the poultry. The egg yolk content is going to increase, the biomass is going to increase, and they also provide the immunity to the poultry if the feed is given with the spirulina, either microalgae. It is early given. It also increases the development of milk. The lactation is increases not only in the animals but to the human also. Those who are feeding mothers, expecting mothers, they are given these algal diets, and they help in improve, you can see, the fertility, the immune system, the skin, and also this reduces anemia and mortality rate, improve the disease resistance. So all kind of things are there. If you look here, you can see the algae as a therapeutic agent that stimulate the release of the stem cells, and they are also stimulate the natural killer cells, and they are the rich source of omega-3 fatty acid, and the phyco and the blue pigment helps in prevention of the inflammation and other those things. So look here, the role of algae as therapeutic agents. There, I have already said that they stopped even the replication of HIV. HIV virus replication has been stopped, has been prevented. There are reports. So it reduces the diabetes, the hypertension, the chemo prevention of the oral cancer. This is the report of Redanna in Central University of Hyderabad, that those who are having the buccal cavity cancer because of the chewing of the tobacco. So the spirulina doses were given. That has really recovered from the buccal cavity cancer. There are a number of reports are there. So you can see that that is also a therapeutic agent for antibody production, and so many things are there. If you look here, you will find that it helps in our regulation of the blood pressure and all those things. Women health, I have told you that increases the lactation in nursing mothers, avoid miscarriages. So a number of therapeutic benefits are there if you give the algae to them. Not only in this area, algae plays important, but in the case of cosmetics, you can see this is spirulina cosmetic, $1 trillion market there, but not in our country. We are not exploiting this market potential using microalgae as a cosmetic product, because after all, it is antioxidant. It has got the property of antibacteria, antivirus, antifungal properties there. So lot of skin problems, the acne problems, and so many things. In Germany, I have seen even the $1 trillion market is there, but we are unable to exploit. Now you can see that if you use this kind of algae as a face mask and other things, you will find a beautiful skin. So I think that a lot of scope is there. Good, hair also is very good as compared to that. And now here I just want to say about the description of the pigment. This is the very important thing. The C-phycocyanin present in blue-green algae, which we have purified in Ash Biotech Labs Private Limited, that if you only purify up to a certain level up here, it is a food grade. You can use as a coloring agent for our food products, and they are safe. They are not the aniline-based dye. If little bit purification is increased, you can use in the cosmetic industry. But when it goes to the analytical grade, the same phycocyanin, it is used as a biomarker for detection of the cancer and for the detection of the heart diseases. And let me tell you that Spirna powder costs around 1,000 to 2,000 rupees per kg. It is available in the country and in China and in our country also. When it is converted into tablets, it becomes around 50 to 40,000 rupees cost. But when this Spirna phycocyanin is extracted and purified at analytical grade, do you know the cost of this phycocyanin powder in international market? 80 lakhs per gram, 80 lakh rupees per gram in international market. So you see the potential of these pigments, but we are unable to exploit and unable to work on that they have got this kind of business opportunity. At the same time, the other allophycocyanin and phycocyanin, you can see phycoerythrin, I was working, the same red pigment is there, having the same property. Now you can see the Dunaliella. This is also a brown algae and the richest source of beta carotene. This is the photograph when I was I went there in Abu Dhabi as a consultant. This is an Al Futaisi Island, a 9 square kilometer island in the Gulf of Aman. And there they are growing this Dunaliella in the brine water, the sea water. You can see that this is the in and around there is sea, and they are growing the Dunaliella, and then they are cultivating Dunaliella for beta carotene. Now, this is the Hematococcus. My student has just submitting the thesis on this one. This Hematococcus is the richest source of astaxanthin. And you can see this is the green alga, and after some time becomes red, and they become the source of astaxanthin, which is also antioxidant and anti-cancer in nature. And so a lot of astaxanthin pigments and tablets are available in the market. Why not to exploit them? Now you can see the biodiesel is also being produced from the microalgae. There are n number of strains which are exploited as a source of bioenergy. Biodiesel, bioethanol, biobutanol, all kind of things are there. And now whole world is emphasizing on the exploitation of microalgae for a source of energy, source of bioenergy. Now, this is again the photograph of my student who has just submitted the thesis on the biodiesel production from chlorella, one of the green algae. This is my laboratory photographs. And this we have developed this facility, outdoor cultivation, and then the conversion of this dry biomass into biodiesel and then analysis. So you can see that they have a lot of potential. If you look here, the corn, if you see that they are producing only 18 gallons per acre, cotton 35, soybean 48, mustard 61, sunflower 102, rapeseed 127, jatropha 202, palm oil 635, algae 10,000 gallons per acre. So they are exploiting the algae as a source of biodiesel, and this is the process how the diesel is extracted, the oil is extracted, because only the non-edible oil can be converted into the biodiesel. That is the government regulation. You cannot convert the edible oil into the biodiesel. That is the provision. But the algae is playing a very important role, and the most of the biodiesel produced from the algae is the better as compared to the diesel, because the after the combustion, the pollutants pollution has been reduced substantially by using that. So we have the lot of potential here. And if you look here, the algae as also source of biofertilizer in agriculture for promoting the nitrogen fixing ability and the providing nitrogen to the plants. So for improving the soil fertility and the crop productivity, algae is exploited as a biofertilizer. Not only this algae, but you can see here this is also the azolla. the water fern azolla, which contains the nitrogen fixing algae, nostoc Anabaena azole in its lower fronts, and that is how the azolla is exploited as a biofertilizer, as a food also, as a feed also, as a various sources. You can see here in the Narmada River 2 years back, a lot of azolla was grown on the surface. The people were saying this is a fungi. A lot of fungi has come in the nature, in the papers also. Then it has been said that this is azolla, it's a water fern, and it can be utilized as a biofertilizer. So, and the Central Rice Research Institute in Cuttack and International Rice Research Institute in Philippines, they have done a lot of work over there on azolla as a food, as a feed, as a cosmetic, as a biofertilizer. Even the IRI they are using, because this azolla contains this nitrogen fixing blue-green algae Anabaena azole, and that provides a lot of nitrogen to the plants. So therefore, they are working as a source of biofertilizer. Now, this is the elements, composition algae on a dry weight basis, whatever the elements are there, they're in the dry weight basis. So now the benefits as a biofertilizer, promotes the growth in the rice agriculture. You are all aware that around 30 to 40% nitrogen demand is fulfilled by the nitrogen fixing blue-green algae when it is used as a biofertilizer. So if the 40% cost is reduced, so our farmers are going to be benefited. At least these are the renewable sources. They are biodegradable in nature. They not only the fixed atmospheric nitrogen, they provide others metabolized to the plants for the growth and survival. Now you can say that increases the soil fertility and the crop productivity, that inhibits the growth of the weed. So many exploitation are there. This is the some of the data from my own old students. They have tested the algae as a source of biofertilizer, not only the rice, but the medicinal and aromatic plants. And if you look here, how the raw algae can restore our planet. That is the bioremediation, whether the algae can be used for the detoxification of the heavy metals, pesticide, insecticide, and even the radionuclide, how the algae can help in reducing the pollution, the few gases, coming out of the industry, how their toxicity can be reduced using as a algal-based photobioreactor. So this is you can see that in Germany and other sources, these power plants are connected to the algal culture. So whatever the flue gases are coming from the chimneys and the industries, they are being circulated among the algal cultures. And then they are absorbing a lot of pollutants from the gases and making them less toxic. And then it is released into the nature. So the day will come when all motorcycles and the cars will have this bioreactor, you can see. Or even when the Delhi-like situation will develop, like by burning the parali, then how we will get the pranavayu? From where we'll get the fresh oxygen? So we'll have to have a algal photobioreactor with us, which can provide the fresh oxygen for to breathe and survive. So that is the solution in that. And you look here, the different kind of photobioreactors. What I'm trying to emphasize here, that lot of photobioreactors are required to be utilized for mass cultivation of microalgae outdoors. We must emphasize the cultivation. We must use algae as a farmer. Because if I'm only working in the laboratory conditions, whatever the data we are generating is for the academic purpose. But there is need that we must cultivate the algae outdoors as a crop. Because if we really grow high density culture, now the global problem is there, greenhouse gases is there, the CO2 level is going to up, the ozone problem is there. Who is going to absorb this carbon dioxide? It is only the higher plants if you go for a lot of plantation, afforestation. And the second is the mass production of ultra density high cultures of microalgae. Because the algae can only absorb, apart from the higher plant, this excessive carbon dioxide. So that is this source in future. That is the solution in future also, that we must grow more and more algae for exploitation, or like Dr. Milind has said that I am using our terrace as a organic cultivation of fruits and vegetables. I am growing for the last 4 years. Whatever the vegetables, green vegetables are required, that is a organic, toxin free, pesticide, insecticide free, no chemical fertilizer. We are utilizing for the last 4 years. There was no problem during this lockdown period. All the vegetables are available on my rooftop. I am requesting everybody, those who are having this terrace, they must utilize this solar energy. What Professor Richmond is the solar biotechnology, how to utilize the freely available solar energy into the dollars, into the biomass. That is important. So therefore, with these few words, I once again thank you to the organizing committee, to the Jadhavji for inviting me for this keynote address. I hope I tried to justify and request to all of our fellow fraternity members, particularly the botanists, the researchers, that please focus on this algal exploitation and mass production, utilization as a source for commercially important products, so that we can develop, we can go for an Atmanirbhar Bharat. So with these words, once again, I am thankful to organizing member, to all of you who have patiently heard my lecture for the 45 minutes or so. Thank you so much. Jai Hind, Jai Bharat.
Thank you, sir. Now I request Professor Shailesh Jadhav, sir, for concluding remark. Shailesh Jadhav, sir. Chairman Shailesh Jadhav, sir.
Thank you so much, sir. First of all, I'm thankful to organizer and other members for giving me opportunity for chair the
Yes, K. Jadhav.
Yeah, chair this session and valuable talk from delivered here by Professor Suren Singh is there, and the
Jadhav sir, आपका वीडियो ऑन करिए ना.
okay, just wait. Yes, now it is clear.
Yes, sir. Proceed, sir.
Yes. So I'm very thankful to all of you, and especially organizing committee to giving me the opportunity for this very knowledgeable lecture by delivered by the Professor Suren Singh from Rani Durgavati University, Jabalpur. Professor Suren Singh delivered is the very important knowledge about the algae on the topic of the algae and human welfare. First of all, he giving the information. Is it okay, sir?
Yes, sir. Yes.
Yes. Giving information that algae is the 70% oxygen in our atmosphere. So it is very important for us. And then he giving information knowledge about the algae as a food. How spirulina is the very important for the protein, source of the protein, and not only protein, but some algae for the beta carotene, some of the antioxidant. So many use for the food purpose or supporting purpose this algae. If you use this algae, then we overcome to the malnutrition problem of our country. He also explained here, also Dilwar also presented here, that not only food, but so many examples of so many other use also algae, like cosmetics, biomarker, biodiesel, bioethanol, biofertilizer, bioremediation. So we can say that this lecture is the very important and very knowledgeable for us. And I am congratulate to Dr. Suren Singh for his wonderful lecture. So thank you so much to all of you, and especially Dr. Suren Singh for giving the very important knowledge for this seminar. And I hope.
Thank you, sir. Thank you. Namaskar, sir.
Thank you. Thank you so much too. Again, thank you. It's okay, sir. Professor Milind Jadhav.
Thank you, Professor Jadhav, sir, for sparing your valuable time for us. I am also grateful to Professor Surendra Singh for a nice, informative, and knowledgeable lecture. Thank you, sir. Thank you very much. Thank you, sir. Now I request Dr. Sangeeta Sutar ma'am to proceed for further session.
Good afternoon. Am I audible?
Yes.
Good afternoon, everyone. Now let us proceed for invited talk, which is to be delivered by Professor K.K. Sahu sir. Professor K.K. Sahu sir is working as Professor and Head, School of Studies in Biotechnology, Pandit Ravi Shankar Shukla University, Raipur, Chhattisgarh. His area of research is study of probiotic traits on seed germination of various crops. He is also studying on effect of salinity and heavy metals on physiology of plants. His research group is also working on synthesis of nanoparticles from selected plant species. 9 students completed MPhil and 7 students completed PhD under his able guidance. At present, 3 students are pursuing PhD research work. Professor Sahu sir has completed 8 major research projects funded by different funding agencies such as UGC, DRDO, CG COS, etc. Sahu sir has published more than 60 research papers in national and international journals. Professor Sahu sir has worked as a chairman, member, and subject expert in different academic and professional bodies. Now I request Professor K.K. Sahu sir to deliver a invited talk. Professor Sahu sir.
So good afternoon to everyone. Thank you very much, Dr. Sangeeta madam, for giving such a nice, wonderful introduction of me. Am I audible to everyone?
Yes, sir.
Yes, sir.
Okay. So thank you, Sangeeta madam, for giving such a nice introduction of me. I am really grateful to you. Moreover, I am also hearty grateful to Professor Milind Jadhav sahab for giving such a wonderful opportunity to share some of my experiences and the research data among you people all. So thank you, sir. Thank you very much for giving this important opportunity. So shall I share my slides to all the people?
Yes, sir. Yes, sir.
Yeah, let me try whether I'll be able to share or not. I have never used this Zoom. I used to use the Google Meet. So I'll try my best to share the slides. So I'm sure that you people might be able to see my slides now. Hello.
Yes, sir. Yes, sir.
Okay. So today I am going to speak something in relation to approaches for diagnosis and management of heavy metal toxicity for improved production of the particularly crops. Okay. So shall I move forward?
Yes, sir.
Yeah, okay. I need some sort of the what response from the audience, because I am not able to see anyone, na, so that I'll confirm that I am audible to the person, or my slides are visible to the person. So I'll ask in between to respond from anyone. Okay.
Okay,
So
sir.
I am moving forward now. So what exactly the heavy metals are, or whom we call the heavy metals? So this heavy metal terms includes only the elements with specific gravity above 5. So whatsoever elements are having their specific gravity above 5, they are all included under the banner of heavy metals. But we the biologists, as you all people and me, frequently use this term for a range of metals and metalloids, which are particularly toxic to the plants. You may be knowing what metalloids are. So we call those elements as metalloids, which are having both metal and non-metal properties. Those we call as the metalloids. Some of the examples I had included here under this heavy metal list are the copper, iron, manganese, zinc, nickel, cobalt, cadmium, arsenic. This arsenic is nothing but a kind of the metalloid. Rest others are the metals. Okay. So where from we get these metals? So we have a number of sources in our environment from which we are getting the release of heavy metals. The very first one is the mining. As we do different kinds of the mining activities, whether it may be related to the coal, gold, whatever the mining activity you iron you can do, you are releasing heavy metals into the environment. Then if we are going for some sort of smelting of the iron ore or any kind of the ore to purify them, you are again releasing the heavy metals into the environment. Then if we are going for any sort of the metallurgical industries or the processes, again you are adding heavy metals into the atmosphere. Then corrosion of the different kinds of the metals are again one of the important source for release of the heavy metals into the different environment. Then when we are disposing the waste into the environment, what in a improper manner, then again we are adding heavy metals into the environment. Then whenever we are burning the fossil fuel with immaterial, whether it is for running our vehicle or it is for any purpose, you are burning the fossil fuel like the petrol, diesel, etc., you are again releasing a good amount of heavy metals into the environment. Then whenever we are adding the pesticides into the agricultural land, agricultural field, or the vegetable sector, or whenever we are adding the chemical fertilizers, again we are adding the release of heavy metals either in the soils environment or the water environment, or wherever we are adding these things. So these are the number of sources which are popularly considered as a potential sources of release of heavy metals into the different ecosystem or the environment, right? So am I right? Am I going right now? You may be able to see now the slides. Am I audible?
Yes, sir.
Yeah, okay. So please respond in between whenever I'll ask for na. So now once the heavy metals are released either into the soil environment or water environment or anywhere, so then those heavy metal has to be taken, or they are all uptaken by the plant system. So we have different kinds of the mechanism available in the plants. If we are talking about the roots, okay, so if we'll go for a kind of the cross section of the roots, so you may be able to hear a number of transporters are available in the cell wall of the root cell. Okay, we have dedicated transporters available, right? For this, if we have transporter for cell, this one, we have zip finger, we have P transporter, we have IRT transport, we have COPT1, FDR. These are the and different kinds of the transporters are available in the cell wall of the roots. And take note that these transporters are dedicated in their nature. It means a single transporter is responsible for transport or uptake of a particular kind of metal ion. It cannot uptake or transport the different metal ions. So we have, that is why I said we have dedicated transporters on the cell wall of the or the in the plasma membrane of the cell. And after their entry from the soil sample, they goes or they sequestered into the vacuole. The very first thing, they accumulated in the vacuole of the cell. If they are eliminated or they excrete from the vacuole, they are then transported into the xylem part of the plant body. From this xylem part, they then translocate to the entire plant body, right? If we'll have a cross section of this leaf, you can see here we have again the accumulation of different kinds of the metals here, which came here by the means of transport of this through this xylem from root to the leaf surface. So this is all about how the different metal ions are uptaken by the plants and how from the roots and then migrate to entire plant body. And for example, I have shown here a cross section of the leaf area. Okay, so now the next slide will show after entering into the plants, these all heavy metals creates a number of different kinds of the problems in the plant. The very first problem we are able to see through naked eye are called as the morphological changes. Under this we have different symptoms which we can observe. Is the wilting, is this nothing but the drying of the plants because of lack of water. We can see the curling in the leaves, which is the rolling kind of the things in the leaves. We are able to see necrotic patches over the leaf blades, which are nothing, which are happening because of the excessive accumulation or deposition of different kinds of the phenolics material. And we can see the brown patches over the leaf, which are called the necrotic patches also. We will be able to see reduction in the number of leaves and the leaf area. So we have the less number of leaves available in the plants. At the same time, if we have the leaves, the size of the leaves become reduced. So these are all the different kinds of the few examples of morphological changes which we are able to see over the plants through the naked eye. Now after this, we have the different kinds of the physiological alterations which are imposed by the uptake of heavy metals. And for example, I had listed here some of the physiological alteration such as the after entry of the heavy metals, it disturbs the uptake and transport of water. So how this is interrupted? Just by blocking the transporters available for uptake of water on the root cells. So we know that in the root cells we have aquaporins, right? So these aquaporins are responsible for transport or uptake of water by the root. So those aquaporins are blocked by these metal ions, and then they doesn't allow the uptake of water into the plants, and then the plant become wilted or the dried. Then these heavy metals causes inhibition in nutrient supply. How? By again blocking the different transporters available on the root cells for uptake of different kinds of the essential nutrients are micro and macronutrients. So what this heavy metal does, they just compete with the different kinds of the nutritive elements, and after competition they occupy their place over their dedicated transporter, and by they doesn't allow the entry of metal ion, right? Like for phosphorus, the arsenic is having more availability for the phosphorus channels. So immediately if arsenic will be there, it binds with the phosphorus channels and doesn't allow the entry of phosphorus. Thereby, there will be a crisis in the ATP. Okay. Then it causes inhibition in stomatal opening. How? By imbalancing the sodium potassium balance over in the guard cells. It causes their closing and doesn't allow the opening of stomata. Lower pigment content. So what heavy metal does after entry into the plant cells? It causes magnesium crisis of the magnesium ion, and if the plant doesn't have magnesium ion, it do not be able to synthesize the chlorophyll. At the same time, this heavy metal causes increase in the levels of chlorophyllase enzyme, and we know that these chlorophyllase enzymes are responsible for degradation of chlorophyll. So two mechanisms are there for this lower pigment content or decline in chlorophyll content, which one is by the crisis of magnesium ion and another is by increased activity of chlorophyllase enzyme. After this, the heavy metals causes reduced growth and productivity just by hindering with the uptake of nutrients, the micro and the micronutrient. Then it causes disruption of membrane. How? It causes release of reactive oxygen species, or it causes what stiffening in the membrane. And we know that the membranes are flexible in nature because of the lipid protein bilayer, and it is a flexible in nature. So as soon the heavy metals enters into the plants, it makes this gel like this flexi membrane structure in the gel form. And if the lipid protein bilayer will become jelly in nature, it will lead the breakage or cracks in the membranes, and from those areas the protoplasm or cytoplasm of the cell will be leached out, and it causes death of the plant. Reduction in the biomass, again by hindering with the uptake of nutrients and the other essential nutrients. So these are all physiological alterations created by heavy metals after their entry into the plants. Now, after that, it causes a number of biochemical changes. The very first one is production of reactive oxygen species. These are nothing but the another toxic or dangerous form of oxygen. We'll see details of this in the later slides. It causes alteration or degradation or glycation or nitrosylation or glycation of the protein. Heavy metal also leads inhibition in the different kinds of the key enzymes which are responsible for different kinds of the metabolic activity of the plants or for defense system of the plant. So these are all got altered because of the entry of heavy metals into the plants. Now, these are some sort of the molecular changes which were reported by different scientists time to time in response to heavy metal stress. The very first one is the strand breaks of the DNA. So it causes breaks in the either single-stranded or double-stranded DNA by different mechanism. It causes mutation in the different sectors or different genes. Then it causes aberrations or damage or deterioration of the chromosome. It hinders the maintenance or it reduces or the length of the telomere, and we know that the telomere are nothing but the cappings available on both the ends of the chromosome, so they are called telomere. And if those ends were disrupted by some means, you may have the problem in your chromosome or the plant's chromosome, and thereafter that will be reflected as an injury symptom. It also causes inhibition of the DNA repair enzyme. We have the plants are having two different mechanisms, type 1 and type 2 of DNA repair. So both those repair system were altered because of this heavy metal by their hindrance with the enzymes responsible for the repair of the DNA. Okay, so these are some of the molecular amendments which are shown by different scientists in response to heavy metal stress. So we have seen different 4 categories of the alterations imposed by heavy metal. The very first one is the morphological changes. Then we have seen the physiological changes. After that, we dealt with the biochemical changes, and now we are finished with the molecular changes. Is it okay till this? If anyone is having any query, they may be posed. I'll try to answer those. Anyone is having any query on this? Can anyone say yes or no?
No, sir.
No, sir.
Okay. Yeah, thank you. So I'm moving forward now. So this is the whole story of the heavy metal stress, okay, which we say the oxidative stress condition inside the plants because of heavy metal. So as soon as the heavy metal enters, it creates synthesis of oversynthesis of reactive oxygen species. These are some of the examples of reactive oxygen species. Now, to take care of this reactive oxygen species, plants are having these kind of defense system, right? So if we have equilibrium in which this antioxidant and the ROS, if these are in the equilibrium, if they are in the proportion, so we have these kind of healthy condition of the plants, right? If this system has become imbalanced, in which terms? Like this, when we have ROS in more amount compared to the antioxidant in any of the plants under any of the stressed condition, will have oxidative stress condition, and plant will look like this. And these are the some of the molecular damages reported during oxidative stress condition, which is deterioration of the lipids, amino acids, proteins, nucleic acid, pigment, and these are some sort of the cellular damages, which are the membrane damage, loss of organelles function, reduction in metabolic efficiencies, reduced carbon fixation, leakage of electrolytes, breaks in the chromatid, mutation. So these all sort of this molecular and cellular damages is responsible for causing these kind of cell death, because of which we'll have reduction in growth of the plant, and ultimately we will lose yield of the crop, right? Now this is another condition. This is a methyl glyoxal. Mg stands for methyl glyoxal. Two different enzymes are responsible for synthesis of this Mg, methyl glyoxal 1, glyoxalase 1 and 2. So if the plants are having normal condition, their levels are little bit under control, and if these plants encounter any kind of the stress condition, for example the heavy metal exposure, there is a excessive accumulation of this methyl glyoxal, which reacts with this protein, DNA and RNA, and again causes synthesis of this superoxide radical or free radical, which again causes increase in the level of ROS. And if it will be happening, we have again this kind of the complete network available. So this is complete story of oxidative stress condition which prevails at the time of exposure of heavy metals into the plants. Okay, now I am moving forward. Okay, so we have in the plants different sites are there where we have production of this reactive oxygen species. A number of sites are there in the plants which are responsible for production of ROS. The very first one is the peroxisome, where we have xanthine oxidase enzyme, and these are the things which are happening in the peroxisome which causes production of superoxide radical. Another site of ROS production is the cell wall. We have cell wall associated peroxidases, diamine oxidases, and if we have imbalance in these enzymes, we have production of ROS. We have the plants are having apoplast, again is responsible for release of ROS. If it has the abiotic stress condition. Then another site is the endoplasmic reticulum, where we have NADPH dependent electron transport system, flavoprotein, cytochrome B5, cytochrome P450. These are all responsible for production of ROS. We have plasma membrane, we have the chloroplast and the mitochondria. These two sites are very potent sites, the mitochondria and the chloroplast, because here the any system runs their electron transport chain, right? And electron transport chain is the most potent mechanism for release of the reactive oxygen species under any kind of the biotic stress, abiotic stress, or disease condition in both plants. And this mitochondria serve as a potential site for production of ROS in the animal system also. So these are the different these are the different potential sites where by which we have production of ROS in the plant cell under any kind of stress condition, right? So now the plants are having different kinds of ROS. Among those, I have listed here the most potent four ROS. So they are the superoxide, right, which are moderately reactive with ROS with 2 to 4 seconds of half-life. Usually first ROS to be generated, सबसे पहले इसी का generation होगा, and then it is nothing but around 1 to 2% of oxygen consumed by the plants leads to the production of superoxide radical. Then this is called singlet oxygen, which is the first excited electronic state of oxygen. See, whenever oxygen is having one unpaired electron in their outer shell, so they are called as the free radicals, right? Because of one pair electron, its tendency is to either it want to have one electron from the surrounding metal or anything, or it donates it want to donate its unpaired electron. So instead of donating its electron, it tries to combine with any kind of the metal ion as a free radical, and it creates problem. Now then we have the hydrogen peroxide, another kind of the toxic material, which is the product of univalent reduction of superoxide. It is moderately reactive ROS with approximate 1 microsecond half-life, involved both in the adaptation and the plant cell death. Then this is the most highly reactive ROS, and it reacts mostly with the DNA. So DNA is always under threat of this hydroxyl radical attack. This is having more lovingness with the DNA molecule. So these are the as an exemplary 4 different ROS popularly generated at the same time, measured or determined or analyzed in the plant tissues under any type of stress condition. Okay. So these are the targets of ROS. Whenever we have over accumulation of ROS, they target the lipids. It causes the oxidation phenomena. It breaks the chains. It targets the protein. It modifies the protein. It makes aggregates of protein. It makes nitrosylation phenomena. It causes the damage, the breakage of the protein. Then DNA, it causes depurination, or it is having cross links are connected, disconnected, and then strain breaks are caused. Now, so these are the control condition. We have balance between the ROS and antioxidant, and this is the stress condition when we have the imbalance between these two, and when our transcription factor gets activated. So this stress responsive machinery will become active and will have the tolerance or acclimation phenomena, so that will have this normal growth of the plant. If not, we'll have this control condition again. Thus this so this, if by any means, if we will be able to this particular thing, we'll have this kind of the happy condition of the plant even in the stress condition. Okay, so abhi tak sab theek hai kya? Do everything is fine till this?
Yes, sir. Yes, sir.
Okay, thank you. So in order to combat against the ROS stress or the ROS, plant cells are having different kinds of the protective mechanism. those protective mechanisms are categorized under two banner, the enzymatic protection mechanism and the non-enzymatic protection mechanism. So under this, we have different enzymes, and some of those important enzymes are superoxide dismutase, which is always called as the first line of defense of the plants or animal system, because as soon as we have overaccumulation of ROS, this is the very first thing which become active to check to control over the ROS. We have the catalase, we have the ascorbate peroxidase, we have glutathione peroxidase, we have glutathione reductase, we have glutathione S transferase, or any several other enzymes are there. And then we, under the non-enzymatic banner, we have proline, which is a very important amino acid, but it is a magical molecule for control of the ROS. We have different kinds of the sugar. We have ascorbic acid, we have glutathione, we have alpha-tocopherol. So other different things are there, and these are called as the protective mechanism of the plants. So if this normal condition become this condition, that is called oxidative stress condition. So all these phenomena we have for this we have published two different reviews in different two journals in respect to arsenic condition, in respect to fluoride stress in the 2016, I suppose. Now, whenever we have imbalance in the plants among the in between the ROS and the antioxidant protection mechanism, we have oxidative stress condition. So after that, some of the exogenous applications or treatments were applied to check to control those oxidative stress condition. Among those, these are some of the molecules like the salicylic acid, brazinoestride, nitric oxide. Now people are using miletonin, glutathione, and several other molecules are there, which are now used as a amelioration strategy to control over the abiotic stress condition of the plants, or particularly to check the disturbances imposed by heavy metals into the plants. Okay. So whenever we have exposure of heavy metals, we will have these kind of the wilted, dried plants. If these plants were applied or treated with some sort of the potential molecules like the salicylic acid, brazinoestride, nitric oxide, melatonin, etc., you may have again these kind of reversal or heavy plantlets out from this to this. So let's see how such kind of the things or the state has been done by this, by use of this potential molecules. Let's see what are the mechanism. So whenever we are applying nitric oxide NO, okay, if you want to apply NO, you need to use the sodium nitroprusside, okay, SNP. That is the most important and popularly used source of NO. So you have to work out the dose to apply in any of the plants. For dedicated plant, you have to work out its dose. Okay, so what NO does in the stressed plant? It enhances the growth and development of a plant. It controls the redox control. It activates the signaling pathway. It scavenges or quenches ROS. Right. So this SNO or SNP alleviates oxidative stress via different mechanism by enhancing the growth, yield, and photosynthesis. It decreases heavy metal accumulation. How? By just blocking the transport of the metal ion through their transporter. It enhances uptake of minerals. How? By eliminating these heavy metals from their the transporters. It upregulates antioxidant enzyme. How? Just by increasing the expression of the genes responsible for this enzyme. It reduces heavy metal induced oxidative stress marker. Then this NO also helps in the synthesis of the mitochondria, so that we'll have more production of energy. It leads synthesis of chlorophyll, so that we'll have happy photosynthesis. It enhances the number of peroxisome. So these are all different mechanisms imposed by NO whenever we apply is over the abiotic stress plants. Okay, we tried to apply this NO in different our research area, and then we have just applied this over the salinity stress plant, And then we also tried to apply this in respect to the arsenic stress, and these two paper were published in 2017, and this one is 2016. Okay. Now, then another molecule is epibrezinolide. It is one of the important member of brezinosteride. You may be now aware that brezinosteride is a very important hormone nowadays. It regulates a different mechanism or different array of metabolic activities in the plants. So whenever we apply this epibrezinolide in any of the abiotic stress plant, what it does, it reduces the level of ROS generation. How? Just by controlling the activity of ROS synthesizing enzymes like NADPH oxidase. It activates different kinds of antioxidant enzymes like the superoxide dismutase, catalase, which are responsible to check over this ROS. It stabilizes protein and the membrane just by maintaining the fluidity of membrane. It stabilizes the membrane so that there should not be any cracks on the membrane, and if cracks will not be there, it won't leak their cellular material. It reduces anyone is saying something?
नाही तिथं त्यालाच करूत ना त्याचे आडवे अँगल मी घरी पडतो.
Sir, can you please mute your mic? The person who is creating noise are requested to please mute her mic. Okay. Then it causes denaturation of enzyme, and then this is the very important phenomena, phytogelatin synthesis. It is imposed by brezinosteride. So phytogelatin are nothing but a very important kind of the protein which are having capacity to chelate with the metal ion, and those chelated metal ions are then sequestered into the vacuoles. So if no metal ions will be available as freely, so it won't cause the problem in the plant. So this is the mechanism of the brezinosteride over the abiotic stress plant. So we just tried to apply this epibrezinolide in our arsenic stress study, and this paper was published in the Plant Physiology and Biochemistry in 2017 again. Now another paper on the use of epibrezinolide over the arsenic phenomena. It was published in the Biology and again in 2017. Now, very important molecule, proline. We know that this is a simple kind of the amino acid, but it's a magical molecule. No molecule is having such a vast number of the positive impact or beneficial impact as the proline is having. Whenever you apply this proline in any of the plants which are exposed to abiotic condition, this proline, what it does, it serves as a source of carbonate nitrogen. We know that under the stress condition, plant is unable to synthesize photosynthase. So this proline work as a source of carbonate nitrogen for providing nutrition to the plant. It stabilizes protein just by managing the ROS level. It enhances chlorophyll content by eliminating or declining the level of chlorophyllase, at the same time making more availability of magnesium for chlorophyll synthesis. It serves as a osmoregulator. We know that the maintenance of hydrated condition inside the plant is very important, otherwise the plant will go under the phenomena of wilting. So this hydration is maintained by this proline just by serving as a osmoregulator. It reduces metal uptake and accumulation, again by blocking the transporters of the metal ion. It reduces their uptake. It activates both the type 1 and type 2 kind of the DNA repair system. It stimulates activity of antioxidant enzymes. It reduces denaturation of enzyme. It scavenges ROS so that thereby reducing the membrane damage and retained cell metabolic status. It donates electron to PS2 photosystem system 2 so that it will generate NADPH and glutathione production to maintain the ROS condition. It helps in the synthesis of phytogelatin for sequestration of metal and detoxification of metal ions. So this proline is a very cheap amino acid, and but it is loaded with n number of properties. Anyone can apply this proline over any kind of the stressed plant. So this we tried to apply this proline in the arsenic stress condition. This paper was published in the Acta Botanica Croatica in 2018. Now, salicylic acid, again a very important molecule, right? So what it does, it whenever you apply salicylic acid, it causes endogenous elevation in the level of salicylic acid. It's a plant growth regulator. It mediates or activates the signal transduction phenomena, which is any very important cascade of reaction. It regulates the expression of different kinds of gene. If the expression of these genes are got upregulated, you will have enhanced or antioxidant system, you will have enhanced availability of osmolytes, and then uptake of nutrient. If you will have all these three conditions, the plant will show abiotic stress tolerance against any kind of the stress condition. Now we tried to apply this salicylic acid and against the arsenic stress, and then this paper was published in Journal of Soil Science and the Plant Nutrition in 2016. Yeah, this is a very good approach. The young researchers must be available of this technique, which we call the gene expression study, right? And is also called as the genomic approach to study heavy metal stress condition. So if you wish to go for this genomic approach, you need to extract the RNA from the plant sample, which is a very tedious phenomena. You may be aware of this. The MSc jo bhi kiye honge, they may be knowing. So you have to extract the RNA first, then you need to go for the synthesis of mRNA, again a very tough and tedious phenomena. Now after mRNA, you need to synthesize complementary DNA or cDNA, again which is a very tough and costly affair. When you have this cDNA available, now two different mechanisms are there by to analyze the expression of the genes. If you have the PCR, you can go for this kind of the assay. You need to have in this Eppendorf tube the cDNA sample, GNTPs, the primers you all already appear and know this, and then the tag polymerase. You take this, go for a kind of the reaction, then this reaction will be loaded in this polymerase chain reaction machine. This we are using of the ABS system. We are using this of what? ABS microsystem. Then after PCR, you need to go for the agarose gel electrophoresis, and in this agarose gel, you will be able to see these kind of the expression of your target gene. Right. So this is the one mechanism of gene expression study, which we call reverse transcriptase PCR. Okay, this is also PCR, but this R stands for the real, not reverse. This is reverse transcriptase PCR. This is real time PCR, which is a costly affair, but it gives very precise data. If you go for want to go for the RT-PCR, you need to have cDNA, DNTPs, the primer, the cyber green, which is a very costly the reagent, and then the tagman, again a costly material. When you make this material and then load your sample into this RT-PCR machine, we have this machine of ABS system, and then after doing your it takes around 2 hours time to run here. You will have these kind of the data available of the amplicons, and these values are called as the what? Relative abundance or relative threshold value. Okay, so this is a complete phenomena is called as the gene expression study. Okay, so we have done these two mechanism in our lab. We have gone for this process also, we have gone for this process also. Let me show the data one by one. So this is the data of real time reverse transcriptase PCR, where we have targeted 4 different enzymes or the genes: superoxide dismutase, catalase, APX, ascorbate peroxidase, and this GST, glutathione S transferase, against the fluoride stress. Okay, and these were our treatment against the fluoride stress. Spermidine, you know, one of the animal or phenolics material, and then the melatonin, both are the animal hormone, but these are having immense property to revert abiotic stress of the plant. So these are their spectrophotometric data of those enzymes, and these are the reverse transcriptase gel picture of those four enzymes where we have got the expression of important genes. That data was published in this journal Plant Growth Regulation, Journal of Plant Growth Regulation, against this fluoride stress. Okay, we have also gone for the real-time PCR, where we get this relative transcript abundance या जिसको delta value भी हम लोग बोलते हैं. These are the of the similar enzyme, and these real-time PCR data was then published in the Chemosphere very recently in 2019, which is having an impact of 566 point. This was the case in the lat toxicity and acid rain toxicity in the Trigonella phonum gratium, the methi. Okay, so this is the real-time data of okay. Then another mechanism of the heavy metal toxicity assessment, which is RAPD, which we generally apply for the what distinction between two different species of any plant. So that RAPD can be applied here for detection of metal toxicity also. We had applied this RAPD in relation to arsenic stress, and here for amelioration we have taken DPI, it is diphenyl iodonium, EBL 24 epibrozinolide, and proline as a reversal material. And here we have got polymorphism. Okay, polymorphism means the number of the different damaged DNA fragments are called polymorphism. So by tracking the polymorphism of DNA, you are able to check the toxicity stage also. Okay, so this RAPD data was published in relation to arsenic again in the Plant Physiology and Biochemistry in 2017. Okay, now यहाँ तक सब ठीक है क्या? Is it okay till this, so that I'll move forward if anyone is having any query? Can anyone say yes or no? Dr. Milind Jadhav, am I audible?
Yes, sir.
Okay. Sir, do I have time? Sir, do I have time?
Sir, within 5 minutes we can.
Okay, let me try fast. So this is
Yes.
the proteomic approach where you can be able to track your toxicity by using. Here we have done 1D which we call simple SDS page, and this is two-dimensional electrophoresis data. This is two-dimensional electrophoresis data where we have did 2D and then that data was published in this particular paper in 2015. Then right, these are the recent developments in the amelioration of heavy metal stress, the use of nanomaterials. These nanomaterials are having relatively more effective in nature. They are environmental friendly and the cost effective also. So we have synthesized silver nanomaterial in our lab, and these are the different picture which is showing that their characterization in the lab. We characterize that we had made the nanomaterial of around 10 nanometer. Then these are the pictures of the fluorescence microscopy which we did for superoxide and hydroxyl radical in the fluorescence microscopy. And then that all those data were published in this Journal of Hazardous Material in 2018. This is a very highly rated journal. Okay, then this is another paper of the use of silica nano particle in the aged seeds. This paper was published last in this just 2020 only. Then after the nanomaterial, now we tried this carbon dot, which are more important than the nanomaterial because they are having high surface to volume ratio. They have ability to penetrate extreme spaces of cells. They are high biochemical reactive. They are better cell viability. They have swift distribution inside the cell. It required less quantity and more effective than the nanomaterial. Okay. Then we applied this carbon dot recently in the arsenic stressed plants of the Cicer retinum, and these data was very recently in the November only 2020 we published in the Plant Physiology and Biochemistry. So this carbon dot provides more important result or more effective than the nanomaterial, the silver nanomaterial which we used in our previous work. So now after that, nowadays people are using this plant growth promoting rhizobia microorganism for heavy metal stress condition. And you can see here, these are the different kinds of the positive impact of the use of plant growth promoting rhizobia. Particularly these are most of the time bacteria, although fungi and other organisms also. So but here we had shown the importance of the plant growth promoting bacteria in the abiotic stress amelioration in the plants. So after looking this impact, we applied one PDF into the this science and engineering ACRB, and we got that PDF. Dr. Neha Pandey worked under this in respect to the arsenic toxicity in the rice plant. So after working with in these 2 years, we have published these two papers against arsenic stress, where we have used the plant growth promoting bacteria for remediation of the arsenic stress in the rice plants. These organisms not only remediate arsenic problem, but also it helps in the growth of the plants. That is why they are called plant growth promoting rhizobacteria. So these are the two papers published out of that project. And then ultimately I would like to thanks my entire team. These are my team members. I have pictures of some of the my lab mates, and then I have names of some of those. This Professor Nathani was my PhD supervisor. He was just retired 2 months back, so I am really grateful to him for making me able to do all this work which I had shown. And these are my all lab mates, are the fellow colleagues for whom I am always grateful, because without their contribution I would not be able to say anything in front of the any kind of the event or to publish any paper at the end. And then I am also grateful to Dr. Milind Jadhav Saab for giving me such an important opportunity. At the same time, all the audience for listening me such a long time patiently. Thank you very much. Thank you. Thank you.
Thank you, sir, for your informative lecture. Now the session is open for discussion. Is there any question?
So yeah, I am very happy. I'll be happy if you have any query to answer those.
I think there is no question.
Yeah.
Once again, thank you, Sahu sir, for very informative lecture. Your lecture is very nice, knowledgeable. Sahu sir has highlighted on effect of heavy metals on toxicity of plants, especially morphological, physiological, and molecular changes in plants. This research work is helpful in the field of agriculture and environmental biotechnology. Once again, thank you, Professor Sahu sir. Thank you very much, sir.
Thank you, ma'am. Thank you. Thank you very much. Thank you.
Dear participants, now it's time for lunch. After lunch break, we'll all meet at sharp 2 o'clock. After next session, all are requested to join in a sharp 2 o'clock. Thank you, one and all. Thank you very much. चला.